control the electrophoretic mobility of a molecule, internal factors as well as external factors, in which we talked about the charge, status, size, shape of the molecule as internal factors affecting electrophoresis. And apart from that, we also talked about the external factors which might control the electrophoretic mobility of the molecule, major of which was the applied electric field. And within applied electric field, how voltage, current, resistance, and associated temperature variations can affect the electrophoretic mobility, that also we consider. So apart from the applied field strength, we also talked about how the buffer conditions and the choice of buffer might reflect upon the electrophoretic mobility of the molecule. As well as we also talked something very briefly about the supporting medium, which might enhance the process of electrophoresis to a certain extent, and that means can give you better resolution if you actually use a medium. But the necessity of incorporation of a stabilizing medium for electrophoresis came much later, when it was understood that there are certain factors which might uh, not be very much favorable in terms of electrophoretic separation when you carry it out without the support medium. So broadly, there were two forms of electrophoresis that were identified, and one form in which you don't incorporate any supporting medium, and that is referred to as free electrophoresis. And the other form that developed more or less at the same times when free electrophoresis uh, being was getting developed. Another form was developed, which was referred to as zone electrophoresis, in which you essentially incorporate stabilizing media to rule out all those drawbacks which are associated with the free electrophoresis process. So within free electrophoresis, there are two basic types of free electrophoresis that were developed. One is called as the microelectrophoresis, about which we talked yesterday to some extent, and tried to understand that what is microelectrophoresis all about and how it is operated. The the instrument that was designed in order to conduct uh, microelectrophoresis is called as the Abramson's microelectrophoretic cell. Nobody uses it these days, but it was developed for a peculiar purpose, and that purpose also we discussed in the last class. In this electrophoretic apparatus, at the central portion, there is something like a slide uh, shaped, what you can say, electrophoretic cell, inside which you carry out electrophoresis of certain microscopically observable components or particles which bear a certain degree of charge. Like, for example, the RBCs and bacterial cells are suspended in a peculiar buffer, and then they are subjected to a particular electric field, and in that particular electric field their migration from one point to the other inside the electrophoretic cell is observed directly under microscope. So this is essentially done in order to estimate what is referred to as the zeta potential of RBCs. And the zeta potential, I gave you an idea about what is zeta potential. Is basically the potential difference between the charges which are there on the surface of any particular cell compared to the charges which are away from the cell surface And the potential difference between that is referred to as the zeta potential, is essentially a way of ascertaining that whether the cells will flock up together or will they stay apart. Okay. So if there is low zeta potential, then there is more chance that there will be aggregation of cells. And aggregation of cells can lead to several medical or clinical manifestations. And you might lead up into blood clot or clogging of blood vessels that can be fatal. So at that time, when Abramson actually devised this method, this was essentially to find out the zeta potential of RBC in order to assess whether RBCs are hale and hearty, and what is the proportion of healthy RBCs inside your system. Now, this had only a limited kind of an application, so it was not able to purify or separate entities which were not, which cannot be observed under the microscope, right? So there was a need to develop another method, and that is called as the Tiselius moving boundary electrophoresis apparatus. This was done by Tiselius, and after it is named. It's basically a kind of a U-tube, not the U-tube that we know about. It is a U-shaped tube structure. Okay, and within that U-shaped tube, you fill it up to a particular level with a buffer, and inside that buffer, there is a mixture of your sample molecules. And when you subject these molecules to a particular electric field, then depending upon their charge and mass status, the molecules tend to migrate under the effect of electric field, and that creates different zones, or you can say different boundaries of the charged molecules which are going to move under the effect of electric field. A simple example is given over here that if you're talking about, say, this is pure buffer, this is also pure buffer, and this portion that you can see is a mixture of three proteins, say X, Y, and Z. And when you carry out, when you turn on the electric field, then there will be some degree of movement of these charged entities. So say suppose the leaders are the X proteins, then there will be essentially X proteins which will get crowded in this area. And obviously this portion of the pure buffer is now getting crowded with X protein molecules. And because of that, there is some degree of change in the, what you can say, in the refractive index of the medium. In order to form what is called as a boundary. Okay. So what is actually done here, how do you trace whether the separation is happening? What you do is that you actually try and find out whether there is any boundary formation. Like for example, that is done by using certain optics, and this is done by interference optics. So there is a detector, there is a laser source, and through this laser source there is a continuous laser that is reaching the detector. Now what happens is that as soon as this portion starts to get crowded with X molecules during the process of electrophoresis, the detector's ability to detect these lasers is slightly altered. And on the basis of that change, that means because of a certain degree of scattering that of the laser light that will be created by the presence of X molecules here, the detector will start to detect a decrease in the intensity. And that's what is a kind of a boundary that you can detect. So imagine now X is mixed up with Y and it also arrives somewhere over here. Then obviously there will be a different degree of scattering that you will see. So if you scan this entire area by using a laser technique, then you can actually find out that to what extent it is populated with X, and then to what extent it is populated with a mixture, and so on and so forth. I hope that this much point is clear about the moving boundary apparatus. Is this much fine? Yes, if you have any doubts, then you can ask me pertaining to this moving boundary apparatus. Yes. Any questions, or you are okay with what was explained till now? Yes. Okay, fine. So now moving ahead from this point, it there are certain things that perhaps was neglected by Tiselius when he was designing the moving boundary apparatus. And these things actually cropped up later, and he started understanding that there are certain faults in the arrangement, and perhaps there will be uh, you will have to upgrade the system in order to make it better, working better for you. So, what were the faults? Let us understand it with a peculiar uh, question that is to be addressed here. Do you need to consider certain things here when you observe this apparatus in order to in order to obtain a better separation? So let us understand it in a schematic, and find out whether we are going to get a good separation, or whether we are not going to get a good separation if we consider this as a situation. First and the foremost is, if you have a population of molecules, say A, B, C, in your sample, and you turn on electrophoresis and say all three of them are differently charged, then will you get a good separation? That's the basic question. What do you think? Will you get a good separation? Yes. So we consider a situation that there are three molecules A, B, C in your suspended in your sample, which is like this, and you turn on the current. Now this blue area, you can imagine that this is the pure buffer, and below pure buffer you have a concentrated solution of A, B, and C mixed up together. Your purpose is to resolve A from B and B from C, so that all three of them are separated from each other. So will you get good separation if this is the arrangement? Can you resolve A, B, and C separate from each other? What's your take? Yes, anyone? Is the question clear? I believe that I have put up the question clearly. Yes. So can you resolve the three from each other? Can you get A separated from B, B separated from C? That all three are separate from each other. Can you get that? Yes or no? I just want your answer. Yes or no? What do you think? What's your take? Okay. Tanaya and Janki says that you can resolve them. Okay, let's say A is, now I'm just taking a call here. A is say positively charged. B is negatively charged. And C is also say positively charged. Now can you separate them? I have just added one perspective here. Okay, I have given you a, a way to think over it. That if I have A with positive charge, B with negative charge, and C with positive charge, can you segregate them from each other? Yes. So essentially, to what end will A move? There is something. Tanya says yes. Okay. Essentially, A will move towards cathode because it's positively charged. So it will tend to move in this direction, and considering this arrow as the direction of movement of A. So all A from here will get concentrated somewhere here, right? Correct. Considering B, B is negatively charged. So all B will it will move towards positive end. So all B from here will try to get concentrated towards this end, correct? Right? Fine. But what about C? Yes. So what about C? What will happen to C according to you? Yes. Any idea? What will happen to C? Will it get separated from B and A? The answer is no. It will not get separated. The reason is I haven't described to you that what is the extent of charge on C, right? I have just told you the type of charge, positive charge, negative charge, positive charge, right? Can I not make it right? Obviously, C will get attracted towards cathode, but A is also getting attracted towards cathode, right? So will you resolve A from C? That's what is the question. It is not going to get separated well unless you know something more about C. So if I say that A has two positive charges, B has a negative charge, and C has one positive charge. Now, can you say this, that A will get separated from B and C? I'm just giving you one step by step a way of looking at it. Janki says yes. There's still some problem here, Janki. The problem is that you may visualize it as a process that, okay, B will get concentrated here and will get migrated to, because it is migrating towards anode. A and C will be migrating towards cathode. So you may imagine that A will form a boundary somewhere over here and will get populated over here, while C will be following A. The problem is you are never going to get complete resolution. One, complete resolution in the sense you will never get A followed by C followed by B situation. You will never get these three in three different zones. The reason is simple. They need to travel, some molecules of A always need to travel from here, from this side of the vessel to this side of the vessel. Same is true for B, which needs to travel from this side of the vessel to this side of the vessel. Now, because C is somewhere in the middle of the two, it will always remain concentrated in this area. So most of the C you will find here. Although you might find B here maximally, so this area might be B, this area might be A, but still there will be always some maximum portion of C over here. Some portion of C will be here concentrated. So basically what I'm trying to convey is that you are never going to get complete resolution. There will be no complete separation of A from B from C. Agree to the point? Okay, and that is what is shown over here in this figure. You can see that there is never complete separation happening. You might get some zones of some molecules, but there will be always a mixture remaining. There will be no complete separation. This is one point. Point number two is, what about those molecules of B which are already here at the beginning of the process? Will they not go and strike anode? By the time all other molecules of B are moving from this end of the tube to this end of the tube, these molecules need to catch up to anode. The path is very small. Yes or no? So will they not strike to anode? And that will defy your purpose of electrophoretic separation, right? Are you getting my point? Same is true for A over here, which is already very near to cathode. So all those molecules of A which were at the beginning of the experiment already here will immediately move towards cathode and will strike cathode. So they will be destroyed. Yes or no? Are you getting the point what I'm trying to convey? Okay. So if that happens, yes, obviously some molecules of B will actually strike anode and will be destroyed. That means you will lose those molecules, along with some molecules of A which will strike cathode and you will destroy those molecules. So that is also going to be happening. One, however, most important or major problem in this process is that when B or C or A are migrating through other molecules, which are their migration will be in opposite directions. Like, say, if I draw cathode over here and anode over here, okay, just for simplicity's sake, instead of the U-tube arrangement, when all the molecules are at the center, and now they are trying to migrate from here to other poles, during the process, they are going to pass through each other, right? So in that process, they will be creating a large degree of friction. Do you agree? There will be a large amount of friction when they are going to move. Imagine now that A is moving from this crowd of B negatively charged molecules and slightly positively charged C molecules, and is trying to reach up to cathode. It's going to pass through all these molecules, the crowd of all other molecules. And during this process, will it not be causing a large amount of friction while moving towards cathode? Yes, what do you think? If there is a large amount of friction, what does that reflect? What is friction going to create in the system? Yes. We talked about this. If there is more friction, then what happens? Yes? Anyone? More friction leads to heat, right? There is more heat production. Now, if there is opposite motion is actually going to create more friction is basically there is more heat production. So the apparatus will start to heat up. Although there is very less chance of evaporation here happening. So the reason why he designed it like this is because he wanted to avoid evaporation of the buffer due to heating. Okay, so there is less chance of evaporation of the buffer, but the apparatus will start to heat up. And if there is more amount of heat generated, then it leads to a process of what is called as setting up convection currents. Do you know what are convection currents? Yes? Anybody knows what are convection currents? And in what way that might be a problem? Convection current is something like this, just for your understanding. Okay. Imagine now that you have a beaker filled with water and you are heating by using a burner. There is, you are trying to heat water. Have you seen some kind of motion of the solvent when it is getting heated? Basically, the bottom molecules which are in direct contact with the source of heat, they get energized first. Energized meaning they get heated first. And they are set into motion. Basically, when they get energized due to heat absorption, they are set into motion. And you might have observed this, that they tend to move away from the source of heat. Okay. What they're actually doing is that they're striking other solvent molecules in the path and trying to lose the amount of energy that they have gained. Okay. When they do that, then this is a continuous process. Okay. And these mobile solvent molecules, which are mobilized due to production of heat or due to being in contact with heat, this process is referred to as convection. So convection currents are basically these set of mobile molecules. So what is actually happening inside the beaker? Inside the beaker, if you visualize, then the molecules are moving away. They get cold enough because they are losing their heat with as they get stuck up with other stable molecules here. And they, once they lose their energy, they come back down and get heated again. And this cycle continues till the entire solution gets to the same temperature value. Okay. Now the problem with this process is it is setting up molecules of solvent in motion. Okay. And that is what is the problem here. Imagine now that because of the friction, there is increase in heat or increase in the temperature of the system. And when the temperature increases, there will be convection currents generated. So the solvent molecules will start to move. And when solvent moves, will you not expect that these solute molecules will also be moving along with the solvent? Because solvent is the medium in which you have suspended your solute molecules. So if there is heat production, will they not be getting energized due to heated solvent molecules, and those heated solvent molecules will strike your solute molecules as well, setting them into motion? Will this not happen? First of all, did you get the idea of convection currents? Yes. So if this, imagine now, apart from the electrophoretic mobility or apart from the electric field being applied and being responsible for the mobility of these molecules in this vessel, there is another force which is acting on the mobility of the molecule, and that is convection, produced because of friction. So now, do you expect good separation in such situation? right? So I hope that the question is clear. So do you expect a very good separation here? Obviously not. You will not get good separation because convection current don't have a direction, right? They will be setting up all the solute molecules in motion because of the solvent molecules energized status, and hence everything will be messed up. So instead of getting a good separation, you will not get what you can say a good isolated bands. One more important aspect is isolation. Although you can see over here that X is separated, XY is still mixed, XYZ is still mixed, XYZ is still mixed, Z is separated. So X is separated, Z is separated. Y hasn't got separated completely. But can you still isolate this X out? Can you take this out and use it for your purpose? Yes? Is this a valid question to put up? First of all, do you think that is valid to ask? By hook or crook, I have got this situation in which I have placed this entire assembly, say for example, in a jacket which is filled up with a coolant. So I am maintaining the temperature, so the problem of friction is gone. I can put it, right? I can put it in a water bath and I can maintain the temperature. By doing so, I will get rid of this convection process. Rather, not get rid of completely, but still, I will minimize convection. And say, suppose you have got this kind of a separation. So can I separate this X now? Can I take it out and use it for my purpose? Do you think that this will work? Yes? Tanaya says yes. Problem is this entire liquid situation. Have you ever tried this? Meaning, imagine that there are two solutions which are kept together. They are miscible with each other, but they are kept together. And one solution is at the top with a very slight layer of that particular solution. If you try to aspirate this, you will be actually aspirating everything out. So instead of getting separated X, you will get a mixture of X and Y. Do you agree? You'll have to aspirate it out, right? There is no kind of an outlet here so that you can collect X, whatever X is getting out. Do you agree with this point? If I put up a pipette over here and try and aspirate it, I will be aspirating all this stuff because once I stop the electric current, this will be again brought to the same situation. The molecules will again try to diffuse in an area. Diffusion, you know, the process. Diffusion is a process of movement of solute molecules from a region of high concentration to a region of low concentration. Now, do you agree that X is highly concentrated in this area, right? It is heavily concentrated with X. So now as soon as I turn off the current that was pulling X towards this positively charged electrode, as soon as I turn it off, then X will start to move down, correct? Are you getting the point? So ultimately, even if you can get separation here, you cannot isolate. You cannot get an isolated band, or you cannot get an isolated solute in such a situation. So realizing all these problems which are associated with Tiselius moving boundary electrophoresis, there was development of another what you can say way of performing electrophoresis that is called a zone electrophoresis, in which it was recognized that if you can incorporate some kind of a stabilizing media so that convection currents are minimized, after separation diffusion is also minimized, If you can work it out, then perhaps it is going to work wonders for you, and you will get a very good separation, and you can preserve that separated sample entities altogether. So considering that, zone electrophoresis was developed, considering the limitations of moving boundary electrophoresis, zone electrophoresis was actually tried out. Okay, so zone electrophoresis is a process in which you are actually trying to get rid of moving boundary electrophoresis, all the problems that we have discussed. So as of now, is it fine that we talk about is it fine that we talk about zone electrophoresis in our next class? Because I believe that this is good enough understanding for you to understand some details about moving boundary electrophoresis. And zone electrophoresis we will talk about in our next class. Is it fine with you? So if there are any questions, you can ask me. And about zone electrophoresis, we will talk about in our